Hi friends, welcome to Infinity World. Today we are going to see about PCR. Polymerase chain reaction is a technique used to amplify multiple copies of a specified sequence of DNA called target DNA. The DNA is collected from a sample of interest. For example, someone's hair follicle, an ancient bone, a plant seed, or cancerous tissue. PCR relies on a thermostable polymerase enzyme. It is called chain reaction because the result of one cycle is used immediately for the next cycle. This allows exponential growth to happen. PCR was invented in 1983 by the American biochemist KARY MULLIS at CETUS Corporation. Principle: the three steps form a cycle that is repeated many times to produce millions of copies of a specific DNA sequence from a small initial sample. PCR amplifies DNA fragments of between 0.1 and 10 kilobase pairs in length. The amount of amplified product is determined by the available substrates in the reaction. A solution containing the target DNA is placed in the machine. This solution contains the target DNA sample, primers, nucleotides, Taq polymerase buffer, and magnesium chloride. The reaction is carried out in a volume of 10 to 200 microliter in small tubes. The process requires a special machine called thermocycler. The thermocycler heats and cools the tube to achieve the temperatures required at each step of the reaction. Steps involved in PCR process. PCR involves the repetition of 3 steps. Step 1. Denaturation. The temperature is increased above 90 degrees. This heating breaks the hydrogen bonds between complementary base pairs, causing the two strands in DNA double helix to come apart or denature. Step 2. Annealing. The temperature of solution is lowered to 50 degrees Celsius. This causes primers to attach to DNA. Primers match specific sequences of DNA. They are needed to start the process of making new DNA. Step 3. Extension. The temperature is again increased to 72 degrees Celsius. Taq polymerase extends the primers, synthesizing new DNA strands. The cycle is repeated for 20 to 30 times to produce millions of copies of a DNA strand. This reaction can be performed within 2 to 3 hours with certain high-speed machines. After PCR has been completed, AGAROSE gel electrophoresis can be used to check the quantity and size of DNA fragments produced. The size of PCR products is determined by comparison with a DNA ladder, a molecular weight marker which contains DNA fragments of known size run on the gel electrophoresis with PCR products. Applications of PCR: direct detection of microorganisms in patient specimens, identification of microorganisms grown in culture, detection of antimicrobial resistance, analysis of genetic fingerprints for DNA profiling in forensic science and paternity testing, gene cloning and manipulation, diagnosis and monitoring of hereditary diseases. Thanks for watching. If you like my video, subscribe and share my YouTube channel.